This is the network pathway annotation tutorial for scGRNdb. Let's get started. So I have a fresh session here, and if you want to see what network pathway annotation entails, it'll annotate your own network with biological pathways and disease gene sets. Your required inputs include a network file, a minimum and maximum module size for detecting neighborhoods in your network, choosing a species that matches your gene set file, and then selecting one or more pathway databases to annotate your network. The analysis will perform a module detection on your network and then find the pathways and disease genes that are enriched for those modules. You can then visualize your own network in the network browser. Great. So let's get started with trying to run the pipeline. As for the network input, here's an example file or example table of what that would look like. It includes a head, tail, and weight, where head and tail are the source and target genes respectively in your network, and weight is some weighted edge, which is optional, but if you have it, if you don't have it, you can also set an arbitrary weight column. You can choose between a human sample network, a mouse sample network, as well as uploading your own file. Here I can upload this Alzheimer's disease microglia network that I have. You can set the minimum and maximum module size. Here I'll set 20, since if I want a slightly larger minimum size module, choose human, and then I can select all of these different databases, pathway databases and disease gene databases that I want to annotate our network with. You can submit your email, submit the form, and then the annotation results will run again. Great, I've run this analysis already in a separate session, and we can go ahead and start looking at that. You can view the details of your form here, and this is what the result table will look like. You can download the modules of your network and download the pathway enrichment of your network here, and again readily view the pathway enrichment table in the browser. So here the pathway will just describe all the enriched pathways, and you can sort by the FDR, or false discovery rate, of the enrichment of these pathways on your modules, if you would like. You can also use these search panes to search for, maybe if you're interested in only DisGeneNet pathways, you can then search and sort accordingly. We can also search for keywords if I'm interested in like Alzheimer's disease. As I mentioned, I use an Alzheimer's disease network. You can see that, yeah, this module 2 seems to be pretty enriched for these Alzheimer's disease genes. So I'll go ahead and visualize that module by clicking this visualize button here. This will again bring me to the network browser, where I can visualize this module 2. I can view the pathways again. This reflects the pathways that are in the pathway enrichment table as well. You can see this Alzheimer's disease late onset in the GWAS catalog. Great. We can also view this module by loading this module here. And this is going to be, yeah, the module that's enriched for these Alzheimer's disease genes. Another thing we can do is search for lists of genes. Let's say I want to look at ApoE, APP, and TREM2 here. These are hallmark Alzheimer's disease genes. I can also search for those here in the search result, as well as increase the search if I want to. Great, now that that's loaded, I want to compare this to a normal microglia, and I can do that by using this compare networks function, which brings us to the table that's like the network browser, where we can choose a network that we want to compare our uploaded module with. So here I want to look for microglia, and I know that the anterior cingulate cortex is in the prefrontal cortex, which is the same tissue as I used in this custom network. So I'll go ahead and visualize that one as well, which will pop up here on the side. I can search for modules here that may also be enriched, like for module 0, for example, we can see it's enriched for similar pathways to that of the module 2. If we look at the biological functions, where we see some GTPase activity also present in this module. And if you want a full list of all of the pathways enriched for these modules, you can always download them and search for the modules that way. So let me go ahead and visualize the module 0 here from the normal or healthy microglia network. And I can directly compare that with the module 2 by clicking the combined networks here. But before I do that, these networks are quite dense, so maybe I'll just focus on the top 15% of nodes in each to remove those that are from the periphery that may not be as important. And then maybe I can decrease this maybe to, let's see what 5% looks like. Yeah, 5% seems to already be a pretty interpretable network. Bring this maybe down to 10%. Refresh the layout. Great, let me perform the combination on this. So this is the most important nodes in both of these modules that explain very similar biological function. And if we zoom in, all these green genes here are shared in those modules. So we can see that there's a lot of shared genes that are known in Alzheimer's disease, like this IMPP5D gene, again, that are all involved in that GTPase activity pathway that we know is dysregulated in microglia in Alzheimer's disease. We can also see some unique mechanisms to the network 1, which is our Alzheimer's disease microglia network, as well as some specific mechanisms to the physiological network in the anterior cingulate cortex microglia. So this is a great way to kind of compare and contrast the dysregulated mechanisms in disease versus normal condition network using the scGRNdb. We can also directly compare if I use the same list of genes, the APOE, APP, and TREM2, and go to the search results here. Maybe I want to increase the search a little bit. And if I combine these two networks, here we see there's a lot more activity in the APOE, APP, and TREM2 in the Alzheimer's disease microglia, which we know to be disease activated, and it introduces a lot of mechanisms that are not captured in the microglia network. So this here is showing that the key Alzheimer's genes, they have a different function or different mechanism in the normal condition that's not captured in the disease network. So again, you can use this combined network function to compare and contrast any disease genes of interest in our networks or your custom networks with the scGRNdb networks.